Hello, everyone. Welcome to this open for comment presentation of the Experiment Metadata Checklist product. This is a product that's been developed within the Experiments Metadata Standard Group over the past 2+ years. We've now reached the first version of that product that we would like to have released as an official product. And therefore, we will walk you through in this presentation what the product is about. So first of all, let's look at the problem statement, why this product has been created. Um, to give a bit of context, modern omics research and clinical pipelines involve many types of assays, producing diverse and complex experimental metadata. So there's lots of pipelines, lots of projects, all producing multi-omics data using sequencing technologies on like whole genome sequencing, RNA-seq, single cell, and then epigenetic assays with the methylome and the ChIP-seq, ATAC-seq, etc. All of these types of assays exist and produce data that's in essence very different from one data set to another. So the problem is that the absence of standardized metadata to consistently describe the differences between these assays undermine the FAIR principles needed to discover the data, to facilitate data reusability. For instance, let's say I obtain two data sets from two sources, and I'm not exactly sure about the experimental conditions in which these were ran. It will be difficult for me to to assess how comparable or how usable together they are. And the third goal is to improve interoperability across G4GH and external efforts. So we'll get back to this, but like potentially how this can be used across different G4GH working groups to improve reusability, interoperability of the data. So the goal of the experiment metadata checklist is to provide a common approach to describing experimental procedures. The version 1 of this product focuses on properties that are common to all sequencing experiments. So we're not looking here into the properties that are specific to transcriptomic data or to whole exome data or these kind of things. Really, properties that characterize data, any type of experiment run on a sequencing platform. So we have a scope statement that we have prepared. I include the link here. It, and it describes well for this version 1 product that we're submitting, what we are aiming to do and what we are not aiming to do. So as part of the data generation workflow, there are lots of, I guess, steps in the data generation pipeline. Like we have cohort and family metadata, and then we have metadata about each individual participants. We have information about the biological samples collected from the participants. These biological samples are being prepared as libraries to be sequenced, and then they are actually sequenced on the machine. And finally, they're being analyzed through bioinformatics analysis pipeline. And what we're looking at here in this checklist is really steps 4 and 5, the library preparation and the actual sequencing on the machine. So we're not looking into information about the biological samples. This is being tackled by other working groups. And once the results are out of the sequencer, like the raw results, the way the data is being analyzed to answer research questions and those things is also out of scope of this checklist. So again, for more detail, I encourage you to look at our scope statement, which is on the GA4GH website. So for the overview of the product, the experimental metadata checklist provides a list of properties for describing the context in which genomic experiments are conducted. So again, a list of properties here means that we're not looking into a full-fledged schema that tells you exactly how to structure the data. This is like a list of properties, a list of things that we feel are important to capture at a minimum to properly describe a sequencing experiment. Some of the key components include information on the assay, specific experimental technique applied to samples. For instance, is this a RNA-seq experiment? Is this a ChIP-seq experiment? Information on the instrument, which platform was used to sequence the library. Is this an Illumina NovaSeq X? Is this Oxford Nanopore, PromethION? Is this a PacBio instrument, etc.? Library information, the library preparation. So like giving a way to describe processes such as extraction, amplification, library preparation prior to sequencing, and informations to about the protocols being followed. And these, this checklist allows you to provide links to versioning systems that would allow you to put a complete description of your laboratory protocol. It's the checklist itself doesn't list all of the properties or the way to organize information about the protocol. The product design draws upon real-world use cases from research consortia and projects such as EINSDC. Like one of our fundamental building block for this was the ENA prototype experiment checklist. So we started from that, and then through months of discussions within the working group, we adapted it, added properties, modified it a little bit to reach consensus. And the International Human Epigenome Consortium had a role to play there, also contributing from their experience characterizing multiple types of sequencing experiments. And this is to ensure practicality and relevance of what we're doing. It's intended to be integrated in the data models, the tooling, and the workflows used by data producers, bioinformatics developers, repository maintainers. So anybody dealing with that sequencing data that can be used and made available in in discovery systems and so on. So we identified the 4 use cases. One of them is repositories and data aggregators. So like resources such as within the INSDC, with the ENA, and other long-term data archivals such as the Pan-Canadian Genome Library, Canada, and so on. We also have large-scale genomic initiatives, like aiming to make their data more accessible, more understandable, and shareable across projects. Like all of these massive data set producers, which are aiming to make their data reusable alongside the ones generated by others. Clinical genomic labs looking for a way to standardize their metadata capture for regulated environments, and digital biobanks characterizing their biosamples with multi-omic approaches. One such example is the BQC19 COVID-19 biobank in Quebec, Canada, which has like 6,000 plus participants, biological samples collected in multiple ways, and then you have whole genome sequencing, you have proteomics, you have metabolomics, you have single cell. There's all sorts of experiments around on the same samples. So this is a kind of example where it's useful to properly describe the data and the experimental data and the way one part of the data set differs from another. Community engagement and adoption. Um, since the beginning, input was sought from collaborators in large-scale genomic projects, such as we identified before, the International Human Epigenome Consortium, which needed a way for better integration with local repositories and modularity in design. The ENA, which supported flexible schema alignment and emphasized ontology mapping. Uh, During the development of the product, like feedback was gradually incorporated to simplify the core model, clarify the documentation, and most importantly, allow for future expansions for emerging technologies. So we're looking here at a field that evolves very fast. So we have a core model here, and then in the future steps, as we'll see toward the end of this presentation, we'll look at ways where we can build additional modules that can build over that core checklist to describe things such as spatial transcriptomics, maybe targeted sequencing methods, and so on. As for engagement and feedback, this checklist was co-developed by multiple community partners who have joined our calls over the past 2 years: IGVF and ENCODE, the SRA, ENA, PHAGE, Biodata Catalyst, the Human Cell Atlas, CRDC, and etc. The checklist is compatible with existing large-scale project dictionaries. For instance, recently we had a interesting discussion with the HCA and the effort that they've been doing on their side to try to standardize the way the experiment metadata is being captured on their side. And it's interestingly, we did the things each on our side, and the conclusion is that like the mapability between the two products is very high, and now we're working on filling the gap and see how we can work together for future steps. As for implementations, early implementers have been closely involved in the development of the checklist. They've been contributing their needs and their concerns at our meeting and in our documentation, the Google Doc documents, the online spreadsheets, and so on. The INSDC instrument and platforms have been mapped to the OMB ontology, and a lot of work in the past 6 months has been done to expand the OB ontology to, uh, to, to fill what could be missing, for instance, in the instrument scheme, like how, like the relation between instruments, like this is an Illumina instrument, and the way it's related to others, yeah. So really looking at all the data that's been submitted up to now in the resources such as the ENA, and trying to fill the gap with the OB ontology, so that like that ontology would be able to be used to properly fill sequencing platform using an ontology term. So we have prioritized capturing the underlying concept in modules, dictionary, rather than enforcing strict nomenclature as a schema. Again, this was a big debate since the beginning, like what kind of product do we aim to ship at first, and there was a lot of discussions in the past few years as well within the Damask group as to like ways to standardize products across G4GH. So we we collaborated with them in to work on that front, and decided that this working group, the experimental metadata standard group, would really focus on the the, the content itself, like what properties are relevant, and once a consensus within G4GH is reached from task and Damask, and then we can work on formalizing this dictionary as a schema. This approach has offered us two advantages. One is to maintain our adaptability and sustainability over time, and ensuring compatibility in future mappings. So looking at implementers, we have first the Pan-Canadian Genome Library. Um, the PCGL is a long-term data archival for genomic data generated in Canada. It's currently being developed, aiming for an initial launch in by the end of this year. Um, from the initial release, the data model within the PCGL has fully integrated the checklist. There's a link to the data model of the PCGL here in the slide. And so far, the experiments metadata checklist has been successfully used to ensure that the most important variables for characterizing sequencing experiments would be captured. We also have a second implementer with the CRDC, the NCI Cancer Research Data Commons. So the CRDC houses the NCI-funded research data, including genomics, proteomics, imaging, epidemiology, clinical and translational studies data. It even includes data such as canine biomedical data for comparative research. Currently, there's a there's multiple data commons within there, each one for each data type with its own submission systems. The CRDC is moving toward a common submission system across all CRDC data commons. Um, the data model behind this common submission system is currently being built, currently being developed, and it has a commitment to integrate the experimental metadata checklist uh, for everything that pertains to sequencing experiments. The CRDC has been working for the past year with the NCI semantics infrastructure team to ensure that NCI common data elements are available for and align with checklist elements. And CRDC is committed to ensuring that we collect data of value to the genomic research community in a way that meets FAIR principles. As for interoperability, um, complements metadata is being collected by other GA4GH standards. So I guess complementary. So like there's multiple metadata standards within G4GH, such as PhenoPacket. There's a data model for Beacon. There's a data model that's being developed right now for QC metrics of sequencing experiments and so on. So we've been ensuring since the beginning that what we're doing does not overlap with other G4GH efforts. No interoperability issues have been identified with existing products. The product could be leveraged by other existing products. For instance, within Beacon, this could be a set of new filters to discover data sets based on any characteristic of an experiment. For instance, I could run a Beacon search using this saying, I'm looking for all bulk sequencing, bulk RNA-seq data for people with a given disease. This could be logged as well to PhenoPacket to link between the data set and the participant biosample. And we're also working with the INSDC and EGA members for future interoperability. As for ethics and security, no identifiable information is captured by the checklist. There's no information on participants, biological samples, genomic data itself. The checklist is compatible with security measures that could be implemented or required in some data sharing system. It's part of a complete metadata breakfast, which means it aligns with other G4GH metadata standards as part of a full metadata compendium. And it enhances provenance and fairness by facilitating data reuse, by improving genomic data discoverability. Implementation roadmap. Is the product implemented and documented? Yes, it's available on the GitHub repo. The link is available here. We have a supporting material, like a working document that has the history of all decisions we've made since the beginning, a documentation of, when I say history, like the decision that the working group, when we met, we've been discussing things, we've taken decisions. This is documented in the working document. We also have documentation on ongoing discussions, such as ontologies. All the agenda in minutes since the beginning are available in another online document. As for the roadmap for adoption, we aim to finalize V1 uh, to well, the product is finalized, but to finalize the product review with the product review committee, getting the approval from the steering committee, it would be our first step, and then approaching driver projects, saying this is a new standard, it's relevant for what you're doing for data sharing. So you like, would you be interested in implementing this, or at least aligning to this with your existing data model? And that might allow us to identify even more things that could be interested to reach in version 2 of the document. And we aim to build capacity via webinars and implementers workshops and so on. The product is of a broad utility. It enables consistent metadata across wet lab and computational pipelines. It bridges data models between genomic consortia, repositories, and clinical labs to facilitate data discovery. Really, like the main goal of this, one of the primary goals, is to facilitate the federated data discovery of these sequencing experiments. As I was mentioning, we've been aligning with tasks and Damask by making sure that the way we produce our documentation and eventually mapping to schemas and those things will be done in a way that's compliant with other GFHH products. Lastly, as for our future plans, what we aim to do from now, tackling categories of assays by partnering with driver projects and other interested parties. For instance, I mentioned the HCA already. We've been recently talking about how we can work together towards this single cell assay category of a new version of the checklist. Deliver user-focused documentation and tooling, such as a, a schema following Damask recommendations, integration of the schema in an upcoming Damask schema registry. We're also interested into formalizing it using JSON LD, engaging with archives that exist to see to establish mappings between them and the checklist and see what they're doing and try to work with them on trying to map between this checklist and what they have currently. Reaching out to journals focused on data set descriptions to gauge their interest in adopting the standard. And lastly, we're aiming for a publication on the product. So that's it for the presentation. I thank you very much for your attention and looking forward to hear comments about the product. Thank you very much.